I revised this set of notes. So if you haven't reprinted them, from page 4 on is revised. So you can print page 4 on if you only want to print the first four pages, yes. So if you want to print from page 4 on, you can. I moved some stuff from another set of notes into this one, and then I revised some things and put some new drugs in there that you guys will be interested in. Okay, so let's start talking about the synapses in general. So what a synapse is in general, you should have learned in anatomy, and hopefully you learned that it is basically the site of communication between a neuron and another cell. If it is a true synapse, it's neuron-to-neuron communication, okay? If it's not a true synapse, buddy, that's so if it's not a true synapse, it's called, a neuron can also communicate with a muscle cell or a glandular cell. So if it is neuron-to-neuron, they call it a true synapse. The other ones are neuromuscular junctions or neuroglandular junctions. And so in the next set of notes after this, we'll talk about neuromuscular junctions. Just so you know, after you learn synaptic function, then a neuromuscular junction basically just makes the postsynaptic cell a different cell. But the synapse works exactly the same. So this will directly correlate to what we're doing in the next set of notes. So today, what we're going to concentrate on is true synapses, so neuron-to-neuron communication. There are actually two different types of synapse. One of them is called an electrical synapse, and just conceptually, even though we're not going to spend a lot of time on this, electrical synapse. If I have two cells that are going to communicate, and I stick them together with a gap junction, can you imagine that I could actually have a continuous action potential between cell A and cell B if they're stuck together with gap junctions? And that's awesome in some ways because it's really fast, right? And a direct communication. But the thing that's not awesome is it means that cell B always has to do exactly what cell A does, right? Does that make sense? So that would be terrible in an adult brain because it means that if I got a single action potential on a single cell, I would get action potentials on every damn cell, which would make you completely batshit crazy. So you definitely do not want all of your brain cells on at the same time. So in an adult, there are no electrical synapses in the brain. They do, of course, exist between cardiac muscle cells and smooth muscle cells, but none in the brain of an adult. In the brain of an adult, instead, we have what we call chemical synapses. And chemical synapses are using chemical messengers to communicate. So this is a chemical synapse. But let's go ahead and draw it a little bit slower so that you can make sure that you understand all the players. So if we're going to have communication between two cells, you're going to have communication between two cells. The site of communication between the two cells, this entire area is called the synapse. Now, because you've had anatomy and you've also had a little physiology, you can probably tell me just by looking at this, even poorly drawn picture, which direction information is flowing between these cells. Yes, exactly. So the information is flowing that direction, and because you know that, now I can ask you which of these is the presynaptic cell and which is the postsynaptic cell. Yes, this is presynaptic. And it's the presynaptic neuron, and this is the postsynaptic neuron. Okay, and that's for this synapse, right? Of course, there's going to be another synapse down here, but you always label it with reference to a single synapse. So that's the presynaptic and the postsynaptic neuron. Let's just get the rest of the players involved. So, of course, this neuron has the axon hillock, right? This is the dendrites in the cell body, the axon hillock. This is the axon, and then when you get to the end of the axon, you are going to run into these branches that always exist at the end. And so let's just draw them a little bit more carefully. They look kind of like red-eyed tree frog fingers, or ET fingers, right? Okay. So these guys right here, the fingers are called axon telodendria. Telo meaning end, dendra meaning fingers, fingers at the end. And then the important player right here is this part. This little swollen part at the end, nobody can decide what it's actually called, but it's either called an axon terminal, a synaptic terminal, a terminal bouton, whatever. But the important thing is, why is it swollen? What's stored there? Yeah, neurotransmitters are stored there, so that's going to be the important part. And they're actually stored in little balls called vesicles, so neurotransmitters. So you have synaptic vesicles stored there. Okay, so now let's get a big functional organization or functional understanding of what's going to go on here. So in the presynaptic neuron and in the postsynaptic neuron, you're going to have communication across this space right here. What's the space called? Synaptic cleft. Okay, logic question for you. What is in the synaptic cleft? It's not empty. What's in there? What? Fluid, exactly. It's interstitial fluid, right? Extracellular fluid, interstitial fluid. So it's not empty, right? There's a fluid for something to diffuse across, which is going to be important because we're going to diffuse across it. So the synaptic cleft. And on the postsynaptic cell, what I'm going to have is on the on the dendrites or the cell body, I'm going to have receptors. Okay. So we'll get to the specific mechanics in just a second. But basically the story here is going to be, upon an action potential, the presynaptic cell will release neurotransmitters into the synaptic cleft, and the postsynaptic cell will have receptors that responds to the neurotransmitters, right? So what we've got here is a pitcher, catcher, a ball, and a mitt, right? That makes sense? So what is the presynaptic cell? This is the pitcher, exactly. What is the ball? The neurotransmitter that is released, okay, neurotransmitter. The catcher is the postsynaptic cell, and the mitt is the receptor. Now, the important things about this is that the pitcher in this metaphor does not have a mitt, okay? So even if the ball comes back, it cannot cause a response in the presynaptic cell because there is no receptor on the presynaptic cell. Does that make sense? So because of that relationship, synaptic transmission is unidirectional. It's one way, okay? So even if the ball comes back, so for instance, some cells will reuptake the neurotransmitter after they release it and it does its job. They can reuptake it back into the presynaptic cell. That's all well and good, but that's more like rolling the ball back to the pitcher, okay? Does that make sense? So you cannot send the transmission backwards. And the biggest reason is that there are no receptors on the presynaptic cell, at least not in the synapse. Okay? Everybody good with that? Okay. So, transmission across the chemical synapse is always one way. So now let's get into the details of how you actually cause this. So the function of a chemical synapse. So the neuron is separated from its neighbor by the synaptic cleft. So concept check questions, see if you can field this one. Why, if I have an action potential that goes down the presynaptic axon, why can the action potential not jump this space? Why can't I have an action potential that jumps the space and starts an action potential in the postsynaptic cell? What? It's not myelin that's the issue. An action potential is an entity that occurs on a what? Yeah, it occurs on a cell membrane, right? So in the absence of a cell membrane, you cannot have an action potential. So the deal is, if I want an action potential on the postsynaptic cell, I have to restart the whole damn story over again. With chemical-gated channels, hitting threshold, voltage-gated channels, the whole thing. So I can't have a continuous action potential unless I have a continuous cell membrane, right? So what I'm going to have to do in the synapse is I'm going to have to basically restart this entire thing. So here's what's going to happen. So the action potential doesn't jump across the cleft. The space is going to be bridged with chemicals. And I know that you know, but tell me, what kind of chemical is this? What kind of chemical messenger? Neurotransmitter. Now, it has to meet two criteria in order to be a neurotransmitter. What are they? Released by a neuron and right next to the cell or the receptor or whatever, right? Okay, so this qualifies as a neurotransmitter. We'll learn a bunch of different neurotransmitters in this set of notes. So when the action potential, so how do you actually release the neurotransmitter? The action potential is going to go all the way to the end of the axon without dissipating, right? Because it's going to open every single voltage-gated sodium channel all the way down the axon. And then when it gets to the end, I'm going to meet a new kind of channel that I didn't tell you guys about yet. And this new kind of channel is a voltage-gated calcium channel. And they are only present at the axon terminal. So does it make sense to you just mechanically that the very last thing to get depolarized would be those voltage-gated calcium channels, right? This depolarization starts down there and goes sequentially all the way down the axon, and so the last thing to be depolarized would be here, the axon terminal. Okay, so the voltage-gated calcium channel is going to open, and it opens in response to depolarization. And then what happens is, let's look back at this figure, hold on. Okay, so putting a little functionality into the image. So we had an action potential that goes all the way down to the axon terminal. Number 2 says you open your voltage-gated calcium channels. And then calcium moves into the presynaptic cell down its electrochemical gradient because it is both higher concentration in the extracellular fluid and it's also negative in the intracellular fluid, right? So calcium will move into the presynaptic cell. Everybody good? Okay. It is the increase in calcium concentration in the axon terminal that is the trigger for exocytosis, as long as you have ATP. And there are mitochondria down here, by the way, so you are going to have ATP. Okay? So it's the increase in intracellular calcium concentration in the presynaptic cell is the trigger for exocytosis. So now I'm going to do exocytosis of the neurotransmitter. Which neurotransmitter? Well, there's a good rule, but apparently we just found exceptions to it. Generally speaking, usually, a single neuron will make, store, and release a single neurotransmitter. So for instance, if this was a serotonin neuron, it will make, store, and release serotonin. By the way, serotonin is generally made up here in the cell body and moved down the axon to be stored in the axon terminal. Is that okay? Okay, now, just because they're always going to find a damn exception, some yahoo went and tried to make it confusing again because they just recently, over the last 5 years, found a couple of neurons that make, store, and release 2 different neurotransmitters. But what we don't understand yet is how the hell it decides which one to release. Because if it's just stored there and it's released via exocytosis, which is what it is, right? So this is exocytosis. When the calcium concentration increases, then you fuse the vesicles with the membrane, and then you do exocytosis. So I don't know how you choose which one to exocytose. I have no idea. But let's just pretend that they all work this way, okay? Makes, stores, and releases a single neurotransmitter. And you do exocytosis of that neurotransmitter. You dump it into the synaptic cleft, but it's microscopically separated from the receptor, right? So it just diffuses straight across. And by the way, most neurotransmitters we will figure out are relatively small and low molecular weight, which makes them diffuse relatively quickly. Even the biggest neurotransmitters are less than 40 amino acids. Big, okay? Does that make sense? So it's going to diffuse across and it's going to find the receptor on the postsynaptic cell membrane. So whatever neurotransmitter it is, and we'll learn a bunch, acetylcholine, GABA, epinephrine, norepinephrine, dopamine, whatever, diffuses across the cleft. And then it is going to bind to a protein receptor. And usually, the protein receptors that we're going to be talking about, at least in this set of notes, most of them are going to be chemical or ligand-gated ion channels, which is how we sort of understood the whole process of starting the action potential in the first place. So now what I'm going to do is just to sort of make it come full circle. Now let's say I release a neurotransmitter that binds to and opens chemical-gated acetylcholine channels. This was acetylcholine. And these are sodium channels. Okay. Then what is it going to do to the postsynaptic cell? What polarize it? So this neurotransmitter binds to and opens chemical-gated sodium channels. What's this going to do to the postsynaptic cell? Depolarize it. If you depolarize it enough that you hit threshold at the axon hillock, then you get another action potential. So it's the same damn story told over and over and over and over again. Although, conceptually, you don't always want to turn on the next cell. Sometimes you want to turn it off, or turn it down. So we'll talk about how that occurs in just a second. So do you understand? So what I actually end up doing is the slowest part of the nervous system is the synapse. Okay. There is a delay that occurs there. It's called synaptic delay, but there's no way in hell that you can notice it because your noticers have the same synaptic delay, right? So synaptic delay is always going to slow down a little bit there. Okay, so I'll show you an animation in just a second. So the neurotransmitter binds to the protein receptors on the cell membrane of the dendrites or the cell body of the postsynaptic neuron. Every once in a while, by the way, you can also form a synapse on the axon of the next cell, but those are really hard to understand. So we're just going to assume that these are the simple ones. So I'll show you this animation. I couldn't get it to work in the last class, but you guys got lucky. Okay, so you guys ready? So the same video that you've seen before, we just didn't pay attention to the first part of it because we hadn't actually done this yet. So now what I'm going to have is this represents an action potential. Of course, they're not putting in all the voltage-gated sodium channels because the important part here is the voltage-gated calcium channels are open, right? Calcium moves in, and then I'm going to do exocytosis of whatever was stored there. In this instance, it's acetylcholine. And acetylcholine is going to bind to and open ligand-gated sodium channels, which is going to what polarize the postsynaptic cell? Depolarize the postsynaptic cell. And if you depolarize it enough and it's a neuron, you can actually get an action potential. Okay, so we don't always depolarize though. So let's go back and talk about that. Okay, so there are synapses that are involve sodium channels, for instance, and those are generally considered excitatory synapses. Does that make sense? Not because they're always going to get to an action potential, but it's always the right direction. So if the neurotransmitter binds to and opens a chemical-gated sodium channel, then that particular synapse is an excitatory synapse, right? It's going the right direction to get to an action potential. So let me show you what that means. So here's one right here. So ignore potassium for a second. I don't know why they made it go to. So the neurotransmitter binds to a chemical-gated sodium channel, and obviously more sodium moves in than potassium moves out, right? And I am depolarizing. So that means I'm excitatory. Okay, so you describe what's happening on the postsynaptic cell as an EPSP, excitatory postsynaptic potential. Okay, just break it down. Excitatory for obvious reasons. Postsynaptic is where it's occurring. And potential because it's a graded potential that's occurring on the postsynaptic cell. So if the chemical messenger opened a sodium channel, it causes depolarization and excitation of the postsynaptic cell. If it hits threshold, you'll get an action potential, but you don't always. So this is considered an excitatory synapse when that occurs. Now, conceptually, it doesn't have to be sodium, right? It can be any positive ion that moves in or any negative ion that moves out. Can cause excitation. But of course, the one that we depend on and we understand really well is sodium. Okay, good? So, but you don't always want to turn the next cell on. Sometimes you want to actively inhibit what the next cell is doing. You want to tell parts of your brain to shut the hell up, right? Anxiety, don't you want to tell parts of your brain to shut the hell up, right? Me just pretty much every damn day. I'm like way crazy. I'm like, just chill out, brain. Because I'm still thinking about that thing that I said to that girl in 7th grade. It was horrible. Why was I that person? Okay, so inhibitory synapse. So what happens with an inhibitory synapse is, let's say, a chemical messenger at a different synapse. It can't be the same one because I can't have the same neurotransmitter bind to a receptor and cause excitation or bind to a receptor and cause inhibition. So now let's just say that this is a different situation and this is not acetylcholine. Let's say this is a dopamine synapse, okay? And dopamine is released and binds to and opens a receptor that is also a chemical-gated potassium channel. That is going to cause a what polarizing change on the postsynaptic cell? Hyperpolarizing. And hyperpolarizing is going the other direction, right? It's going the other direction, right? So this would inhibit the postsynaptic cell. Does that make sense to you? So it would make it less likely that it was going to get an action potential, right? Because now instead of going from -70 to -55, I'm going to -70 to -85, right? I don't know what the hell's going on there. Okay, so this synapse would therefore be considered inhibitory, right? And you would describe what happened on the postsynaptic cell as an IPSP, inhibitory postsynaptic potential. Good? Okay, so this is all going on in your brain right now. You are exciting some neurons and you are actively inhibiting others because you sure as heck don't want them all on at the same time. You definitely do not. Okay, so by the way, some inhibitory synapses use chloride channels instead of potassium channels because chloride moves the way you want it to. Okay, so let's make this make sense with the clinical connection. With the clinical connection, epilepsy, because of genetics, injury, whatever, it's a disorder in which you have abnormal action potentials, and sometimes a storm of abnormal action potentials that goes straight across your corpus callosum and causes a big freaking brainstorm that can lead to a muscular storm. One of the first tier treatments for epilepsy, it's not all of the drugs are not based this way, but one of the first tier treatments is to treat with a drug that slightly what polarizes your neurons? Yeah, it slightly hyperpolarizes your neurons. Does that make sense to you? So if you treat with a drug that slightly hyperpolarizes your neurons, then you'll be less likely to have seizures. And one of those drugs, by the way, is primidone. But you're also less likely to have appropriate action potentials. So you can probably understand why people don't like being on those drugs. So I get into a stage in which I'm having more seizures than I want to. And then I go on this medication. But it makes me mentally and physically lethargic, right? I'm dumb. And then I start to gain weight, right? I'm dumb and now I'm fat, right? I like to be clever and hot. That's ideal. And then I go off the drug, right? And then I start having seizures again, right? And then I go back on the drug. And then I stop having seizures, but I'm stupid, right? So this cycle happens over and over. And you can at least empathize with the cycle, right? You can at least see, yes, I understand that my physical well-being is taken care of, but my mental well-being is not in that particular instance. Okay, so now, there is another type of receptor. By the way, all of these receptors that are ion channels, they generally refer to them as ionotropic receptors. Ionotropic receptors. But there is another kind of receptor, and I'm just going to tell you about its existence right now. And then later on, I will show you how it works. This one is called a metabotropic receptor. And a metabotropic receptor is going to be way slower to accomplish a similar thing. It's still going to hyperpolarize or depolarize, but the best metaphor I can think of is an ionotropic receptor. When you're trying to hyperpolarize or depolarize and you do it with an ion channel, it's like making a direct pull shot into the pocket, okay? But a metabotropic receptor uses 5 different proteins to accomplish the same thing. So it's like making a really, really fancy combo shot. You hit 5 different proteins on the way to opening or closing an ion channel. Okay, so it's going to be slower, it's going to be more complicated. I don't know how the hell they evolved. It confuses the hell out of me. And they are like the biggest group of receptors other than ion channel receptors. And we'll learn about them a little bit later. They just tend to act a little slower than ionotropic receptors. Okay, so now I'm going to generate a new graded potential on the next cell. It can be depolarizing or hyperpolarizing, excitatory or inhibitory. But even just as important as generating what I wanted on the postsynaptic cell is getting rid of the neurotransmitter. Okay, so let me see if this makes sense to you. If I release a neurotransmitter and then just leave it in the synapse, then can you see that the postsynaptic cell is therefore insensitive to frequency stimulation? If I leave it in the synapse, do you see what I mean? Okay, so what I need to do in order for the postsynaptic cell to respond to frequency as intensity is that I need to release it and then get rid of it so that I can release it again, right? So that having a constant amount in there means that I was trying really hard to get a constant amount in there. So what I'm going to have to do is immediately after release and causing the postsynaptic cell response, I have to do removal of the neurotransmitter. And there's 4 different ways that you do removal of the neurotransmitter, and they're usually synapse specific. Like some synapses use one, some synapses use the other. So first way is just the simplest way. Just let it diffuse away. That's number 8 that you're seeing right there. Okay, that is fine as long as you're really not dependent on a lot of frequency stimulation, so that you can just let it diffuse away. The next way is you can have an enzyme right here on number 6 that is usually present on the postsynaptic cell membrane, and what the enzyme does is changes the shape of the neurotransmitter so that it can't activate the receptor anymore, right? It can either completely break it down or it can just change a bond so that it actually doesn't interact anymore. But regardless, this enzyme on the postsynaptic cell membrane changes the shape. So for instance, at most acetylcholine synapses, there is an enzyme called acetylcholine esterase, which just pops an ester group off, and then it doesn't activate the receptor any longer, okay? Is that okay? So you can let it diffuse away, you can change the shape of the neurotransmitter so that it doesn't cause the response again. There's a third way that isn't shown here because we have only known about it for a few years, but it's kind of cool. Remember learning about astrocytes in anatomy? Astrocytes are like these sort of structural glial cells that really like help neurons. One of the ways apparently they help is they act like kind of like a ball boy. So if it causes the response you want, the astrocyte will scoop up the neurotransmitter out of the synapse and hand it back to the presynaptic cell for reuse, right? So it's scooping up all the balls and handing them back. It's not causing a response on the presynaptic cell, it's just gathering them and handing them back. So that was not on the figure. And then the one that I bet you've heard of before is reuptake directly by the presynaptic cell. Do you see this reuptake mechanism? It's usually a carrier protein, not always, but usually. And so what this one does is it reuptakes the neurotransmitter, repackages it, right, so that you can use it again, okay? The reason you might have heard of reuptake is, of course, the most common category of antidepressants are selective serotonin reuptake inhibitors, SSRIs. So let's just talk about that conceptually. Let's imagine for a second that this is a serotonin synapse in the central nervous system. Serotonin has tons of functions. One of them is it's a mood regulator. So the way to think about how mood regulation works is if I get a flat on the way to work and my serotonin levels are really low, I'm in tears and considering suicide, right? Or shooting someone on the side of the road. If I have adequate levels of serotonin, I just treat it like any other sucky part of any other sucky day, right? It's just like, that sucked. So serotonin is like a mood regulating chemical, but it's not just that. It also works in other areas of your brain involved with hunger and sex drive. And if you get too much of it, you will hallucinate like crazy because all of our hallucinogenic drugs, save one, are serotonin based. So can you understand why you would want it to be very selective about which synapses it works on? Preferably not the ones that kill your sex drive and not the ones that make you hallucinate, but just the ones that make you be able to chill out, okay? So what it does is, if I have inadequate low levels of serotonin, for instance, I can have anxiety or depression, okay? So I want to get my serotonin levels up. Well, there are some natural ways to do that, but they're not as quick as taking a pill, so let's do it this way. What I do is I inhibit the reuptake of serotonin at selected synapses, right? And what does that do? It mimics the effect of releasing more, doesn't it? Right? You can't make yourself release more very easily, but what you can do is you can inhibit the reuptakes. So for every release of serotonin, you get more postsynaptic cell response. So Paxil, Prozac, Zoloft, Lexapro, more, truly it is actually true that there are more antidepressant prescriptions in this state per capita than any other state. Because you guys need a cocktail. That's what it is. Okay. This just fixes a world of ills. Okay, so that is the reuptake mechanism, and we are going to talk about reuptake chemicals. There are other antidepressants that work with dopamine and norepinephrine. We'll talk about those too. Okay, so you got to get rid of it. If you didn't get rid of it, then you are never going to be able to tell whether you released it once or 20 times, right? So you will destroy your frequency coding if you don't get rid of it. So that should make conceptual sense to you. Okay. So now let's talk about what we call neural integration. So I've been sort of implying that there is a one-to-one relationship between presynaptic and postsynaptic cells, one pre to one post, but that's actually not true. In the central nervous system, it is almost never one-to-one. It's more like 15,000 to 1 in the central nervous system, which makes it really hard to talk about. But once you learn what happens at an individual synapse, you can broaden it and start talking about how all of the synapses would work collectively. So this concept is what we call neural integration. The relationship of pre to post is almost never one-to-one. So what if I have a whole bunch of pre and one post? That relationship is called convergence. And that should make sense to you, right? You converge all on one, okay? So why would you ever want convergence? Well, here's a good example of why you would want convergence. Your hypothalamus gathers data, input, from a bunch of different locations to decide what to do, maybe with your metabolism, right? So it's gathering light data to tell you when it's light versus dark. It's gathering temperature data. It's gathering metabolic data. And it's going to converge on these hypothalamus cells, and they will decide, not consciously, whether to send action potentials or not based on all of these different inputs, right? That makes sense? So that's the concept of convergence. And then you can also have divergence. And divergence is, although each neuron only ever has one axon, there is no limit to the number of times it can branch. So this is one axon with multiple branches. This allows this neuron to be the overlord of all of these, right? So this neuron is going to control simultaneously the action of a whole bunch of different neurons. That makes sense? The hypothalamus also does divergence. There's a bunch of different places in your brain that do divergence. Or another example is the concept of a motor unit. I don't know if you guys learned about this in anatomy, but one motor neuron can control up to 500 skeletal muscle fibers simultaneously. And that's a good example of divergence. Is that okay? So convergence and divergence. One little tidbit that is really interesting is that although for all practical purposes we don't get very many new neurons after fetal development, they used to say none, and then they figured out, of course, there's always an exception. So you don't get very many new neurons, almost none, after fetal development. There is no limit to synapse formation. You are forming synapses. I know that you are because you were in college and you were actually stressing and stretching your capacity mentally. So what actually happens is I want you to imagine that at one point it was just this neuron connected to this one. And then during the process of learning and memory formation, what you actually do is form new synapses. So the neurons already exist. One way to think about it is making connections, right? So I learned something in chemistry, I learned something in anatomy, and now I'm learning something in physiology. 3 separate packets of information, right? Represented by collections, not single neurons in your brain. And so what you actually do when you start to make connections is you actually physically make connections. You can actually form a new synapse by growing a new axon collateral, a new branch off of your axon, and it will make connections. Or what if you really start to understand something, you can actually reinforce the connection that exists by actually making more connections, right? So there is a direct correlation between the concept of memory formation and the concept of synapse formation. It's not that a new synapse equals a memory, but it's that new synapses equal memory. So you are changing the structure of your brain by continuing to learn. And fascinatingly, that means that you end up getting more white matter in your brain. It's phenomenal at doing it when you're a kid, and it gets worse and worse as you age. You know that, right? Okay, and then the other cool thing about that is the best indication of when this occurs very readily is it seems to be occurring, your synaptic formation and your new memory formation seems to be occurring most rapidly during sleep. And there's a couple of branches of research that sort of converged on this. One of them is that they could never figure out why the metabolic activity of a sleeping brain and a waking brain are so similar. Like you use almost as much oxygen and nutrients when you're asleep as you do when you're awake. But of course your sensory input is almost completely cut off. So what the hell is your brain doing? Well, one of the things that they figured out is that it seems that the consolidation of all your experiential memories or all your experiences that go on during the day occurs when you cut off the sensory input. So one of the neurophysiologists that I like, one of my favorites, describes it this way. When you are awake, you are just data input is piling onto you. Sensory input, just everything coming from everywhere. You don't do very much processing of that sensory input when you're awake, but when you go to sleep and you cut off the sensory input, you start consolidating and organizing your information. And one of the other supporting sort of lines of inquiry that supported this is that they couldn't figure out, and still are working on it, they couldn't figure out the relationship between sleep disturbances and memory disturbances. But they're really actually well documented. For instance, Alzheimer's and other forms of senile dementia usually manifest as sleep disturbances before they manifest as memory disturbances. And so they're like, which one's the chicken and which one's the egg? Is the Alzheimer's causing the sleep disturbance? Is the sleep disturbance causing the Alzheimer's? And the answer is we don't know, but maybe it's reinforcing. But we do know that when you don't sleep, especially when you don't sleep well, you have problems with memory consolidation, especially short-term to long-term memory. So it seems as if most of this is happening while you sleep. And it also matters. I mean, you've experienced it. You've been a human. You've been stressed out. You've not had sleep. You probably know how poorly you learn complexities when you're not sleeping adequately. The other thing that's really interesting and important is that they figured out that sleep medications don't let you get the same quality of sleep, and so tend to have a long-term effect on your memory. So Ambien every night for 20 years. It's better than not sleeping, because not sleeping will kill you dead after a while, but you actually don't get the same degrees of sleep. And it seems as if most of the memory consolidation might be going on during certain regions of sleep and not others, and you don't sleep the same way on drugs. Go ahead.
What about something as simple as Benadryl?
I do not know how that one impacts, but I bet the quality of sleep is impacted. But it's not a single night that it ends up affecting you. But most of the people I know who take Ambien every damn night. Like my husband's whole family, none of them sleep after age 30. Like none of them. Like crazy people not sleeping. And bipolar runs in his family, so I'm like, you go to sleep. You just sleep. Sweetheart, you sleep. There's a cocktail and an Ambien. No, I don't do that.
So I know there's not much you can do about Alzheimer's, but if they notice that you're having those kinds of problems, do you take Ambien stuff to kind of like offset getting Alzheimer's, or is it completely unpreventable?
I don't know whether they actually, I don't think that it would delay the onset of Alzheimer's. Because, I mean, it's not as simple as sleep causes Alzheimer's, or lack of sleep causes Alzheimer's, because there's also amyloid plaques that are building up on your brain. But there's a relationship between the three things. I don't think that that's a treatment, but it's actually not in my knowledge, so I don't know. Okay, so anyway, new research. Synapses form during sleep, so make sure you sleep. Okay. Now, let's start talking about how you get this postsynaptic cell to an action potential. So what is not generally true is this. If I get an action potential in the presynaptic cell, I get an action potential in the postsynaptic cell. No, it's usually more than one action potential in the presynaptic cell to get an action potential in the postsynaptic cell. So what you end up having to do is sort of sum what's happening in the synapse. And by sum, I mean if I release 10 molecules of acetylcholine and it caused a small depolarization, then I might need to release 10 more to cause a larger depolarization to try to actually get to threshold. Does that make sense? So there's two different ways that you can do this summation. Okay, so many action potentials are generally required on the pre to get enough neurotransmitter to cause an action potential on the post. And this concept is called summation. And summation is not instantaneous. So here's the delay that I was talking about. The slowest part of your nervous system is the synapse. It's called synaptic delay. It's generally, depending on whether it's a fast or a slow synapse, anywhere between 0.2 of a millisecond to 5 milliseconds, but you have no capacity to notice it at all. So there are two ways to accomplish it, two different types of summation. So here they are. Okay, so first with this figure, I only want you to look at the top graph and really ignore B and C. So what I'm going to do is I'm going to have one presynaptic cell and one postsynaptic cell. The color is trying to tell us that these are both excitatory synapses and this one's an inhibitory synapse. Okay, that makes sense? So this one might bind to and open a what kind of channel? Sodium. Okay, so let's hold on to that for a second. Okay, so if what I do is I release a neurotransmitter that binds to and opens a sodium channel, I get a depolarization, right? If I let it completely repolarize back to resting membrane potential and then do it again, I don't get any summation, right? But what if I do it more frequently, right? What if I stimulate it, don't let it repolarize, and then stimulate it again? Or what if I go really quickly, right? Then what happens is I am getting summation, and they call it temporal summation, because in addition to the bone and the muscle, what does the word temporal mean? time, exactly. So this is a frequency summation. So one pre, one post to stimulate it more frequently. Now, I know some of you guys start thinking, well, what about the refractory period? You don't get a refractory period until you're in an action potential. This is still a graded, right? Refractory periods are only once you involve the voltage-gated sodium channels, right? Okay, that makes sense? Okay, so this is temporal summation, and it only requires one pre and one post. Anybody have any questions about that one? Okay, and then spatial summation is going to involve more than one pre and one post. So let's look at spatial summation. So if I stimulate A by itself, I get that much depolarization. If I stimulate B by itself, I get that much depolarization. If I stimulate C by itself, I get hyperpolarization, right? But what if I stimulate A and B simultaneously, those two together? Do you see that that's another way that I could depolarize, right? Because it really doesn't matter to the axon hillock how you got depolarized. Whether it was 2 different locations, 10 different locations, or 1 location, doesn't make any difference. Because what the axon hillock is doing is integrating or summing everything, right? If I depolarize some places and hyperpolarize, it doesn't matter. If you get to minus 55, you get there. So if I did both of those simultaneously, then I can get to threshold. That's called spatial summation because it's involving more than one place or space, right? So one of them's a time summation and the other one's a space summation. Okay, but can I sum to 0 or sum to no change? What if I did A and C simultaneously? Is it possible that I could actually get no change whatsoever? Yeah, so you can sum to 0. We'll talk about that, how that happens, or why that happens in just a second. Okay, so concept check is what is the fastest possible way that you could imagine that you could get this postsynaptic cell to an action potential? No, but in this situation, this particular situation. Spatial and spatial very frequently, right? So there's no rule that says you can't do A frequently and B frequently simultaneously, right? Then you could get the postsynaptic cell to threshold very, very quickly. Good? Okay, so let's talk about this little situation right here. Why the hell would you ever want to do that, sum to 0? Well, because sometimes some inputs have a desire for a neuron and the other ones have a different desire for the neuron. So talk about, like, did we talk about waking up in the morning? Okay, so let's talk about this stuff. Okay, so when I'm trying to get my ass out of bed in the morning, I haven't had enough sleep, right? So I have 2 different competing inputs. One of them says, you're exhausted, go back to bed. And the other one says, you have a job and you want to keep it. And your children are going to be late to school and everybody will know that you are a mess up, right? So these 2 different inputs. So if I'm trying to wake up in the morning, one of them is saying inhibit, inhibit, inhibit. Don't start this metabolism increasing. You need to go to sleep. And the other one is saying get your crap together, right? So I've got 2 different inputs. Which one wins? Most of the time it's whichever one's the most persistent, right? So one of them is going to inhibit, 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 and the other one's going to excite, 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 right? And if you set your freaking alarm, and if you're like me, if it's going to be a rough morning, I set, not the snooze, I set 6 alarms in a row. They're 7 minutes apart. Like, you know, because you can't just like hit the snooze. So the persistent input of excitation will eventually get me depolarization, right? So sometimes when you are trying to change activities in neurons, there's different inputs that are like desiring different things. And generally it's whatever input is most persistent that will win. Good? Okay, because the axon hillock does not care how you got there. It just cares that you got there. Okay, so temporal and spatial. Okay, so now let's talk about messing with your synapses. Because that's half of what we do in life is mess with our synapses. So with our synapses, especially our C and S synapses, the reason this stuff is fascinating to me is that my C and S synapses are the whole reason that you are smart versus dumb, the reason that you have ADD, the reason you have autism, the reason that you are happy, depressed, anxious, intelligent. All of that happens at the synapses. And your synapses are, you didn't know it, but they are your favorite thing because they are the reason you have favorite things. These are your happy chemicals. These are pretty much the only 3 things you like and you just don't even know it. serotonin, norepinephrine, and dopamine. So not surprisingly, we do a lot of activities to get synapses going, whether it's having sex with someone, jumping out of a plane, snorting something, doesn't make any difference. It's primarily just manipulation of what's going on with your synapses. So there are multiple ways that you can manipulate what's going on with your synapses, and we'll come back to this when we talk about these 3 neurotransmitters. So a bunch of different potential ways that you can manipulate this. So let's just talk about them in theory. So let's say I wanted more of a neurotransmitter. Well, can I get the presynaptic cell to just make more of it? Well, usually not very easily. So if I said, I want to be happier, I want some more serotonin, and I want some more dopamine, okay? Do I have any drugs that can effectively manage the level of serotonin and dopamine that are produced? No. Intracellular processes are very difficult to deal with, and even if you could deal with it, you would only want to deal with them in the regions of your brain related to happiness, and not, for instance, with dopamine in the regions of your brain related to motor activity, or with serotonin in the regions of your brain related to hallucinations. So you can't generally do that with chemical mechanisms. However, you can do it, right? I mean, it's just like a long-term, so how do you get more serotonin? Well, you sleep more, you exercise more, you eat better, you have sex, right? But people are like, well, where's the pill, right? Because all that's like a lot, right? Like in theory, I'm supposed to be doing all that all the time anyway, so give me the pill. So you generally have trouble with interfering with the synthesis of the neurotransmitter. Sometimes there are some drugs that interfere with the release of the neurotransmitter. We'll talk about that in just a second. But even more common is to interfere with the presence of the neurotransmitter in the synapse. So a lot of times, so let's say I want more serotonin and I don't know how to make myself, or I don't want to go through all of that rigmarole to make myself make more serotonin. So I say, hand me the pill. And the pill is quite often inhibiting the reuptake of the serotonin that's released. So it mimics the effect of more serotonin. There are other drugs that do it with different neurotransmitters. For instance, Wellbutrin or bupropion is another antidepressant that works on the other 2 happy chemicals. It's an SDRI and an SDNRI, selective dopamine and norepinephrine reuptake inhibitor. So it works on different synapses and lets those stick in the synapse longer. So you can inhibit the reuptake. Mdma Ecstasy. So let's talk about MDMA. MDMA has a lot of overlap between SSRIs and MDMA. So let's talk about club use of MDMA, and then later on when we talk about serotonin, we'll talk about therapeutic and pharmaceutical use of MDMA. So with club use of MDMA, what happens is MDMA does 2 things. It causes a massive release of serotonin, okay, which makes you feel amazing, and also simultaneously inhibits reuptake of serotonin. So you dumped all of it into your synapses and you weren't selective about it. So if you have enough of it, you can hallucinate a little bit. You're definitely really happy, right, for a really long time. It also causes a release of oxytocin, so you really like people a lot, although just so you know, you actually don't like them that much. Just wait a couple of days. So dump it all into the synapse and then also inhibit reuptake into the presynaptic cell. So you get this massive dose. And that is really similar to SSRIs, but it's like an SSRI on drugs, really, just massive amounts of them. The problem, of course, with MDMA and club use, one of the problems is that if you do a massive dump of serotonin and you dump all of it, then let's say that's Saturday, then on Tuesday, you're going to have the worst damn day of your life, right? They call it Suicide Tuesday, right? Because you are not going to be able to regulate your emotions at all. Do not get into any serious conversations on Tuesday because your serotonin has all been depleted. You dumped it all, and then you're going to have to wait to remake it. Go ahead.
So I'm confused. It sounds like I would think that reuptake would be a good thing because it's like you're recycling it, kind of. But it sounds like the more you inhibit the reuptake, then the more you feel the effects of the.
Well, the effect happens at the synapse, not in the presynaptic cell, right? So that might be a good long-term solution, is more recycling. But I'm really interested in the postsynaptic cell response and not the presynaptic cell activity. So what I'm doing is if I release it and then don't reuptake it, it's almost like I'm releasing it multiple times. Do you see? And by the way, you don't inhibit the reuptake forever. You make the reuptake much less efficient. Is what you do, right? Go ahead.
So does that mean that there are more receptors for it, or like is it just like bombarding receptors?
Are we talking about MDMA or SSRIs?
MDMA.
Okay, so MDMA, regular use of MDMA will downregulate your serotonin receptors really massively. And if you thought you were sad before you started taking it, you are going to be a whole new brand of sad. So when they use MDMA therapeutically, you don't use it any more frequently than once about every 4 weeks.
So then when it's not being
You have to remake it.
taken,
Uh-huh.
so you're just remaking it, or where does like the exit go?
Oh, it just diffuses out.
Oh, okay.
Yeah, so you dump it, it eventually diffuses out, you don't reuptake it, and then you have to remake it. So yeah, you basically cause massive downregulation of your serotonin synapses by dumping them over and over again. I don't know if you guys have ever known anybody who does a lot of MDMA, but you can get yourself a serious chemical-induced depression that is really, really chronic. Go ahead.
Isn't that the same thing as like other stimulants like methamphetamine?
Yeah, meth doesn't work on these kinds of synapses, but we'll talk about meth before a little bit later. Does that make sense to you? So that's the reuptake. There's also a drug or set of antidepressants called MAOIs, monoamine oxidase inhibitors, and what they do is inhibit this enzyme right here that breaks down norepinephrine and dopamine, and they're really effective antidepressants. But you almost never see people on them because they interact with all kinds of like over-the-counter things, over-the-counter medications, and they Nardil and Marplan are these two. And you can't like eat or drink certain things. Like there are certain fruits and vegetables that you can't eat or drink. You can't eat cheese. The hell, that alone would make me depressed. And you can't drink wine. That's half the reason I'm happy, those two things right there. So most people don't take those unless it's inpatient. Go ahead.
Sorry, I didn't understand how that was happening. Those are break. What's that thing?
This is the enzyme that breaks down the neurotransmitters at different synapses, dopamine and norepinephrine. So MAOIs, monoamine oxidase inhibitors, inhibit this enzyme. So norepinephrine and dopamine end up staying in the synapses longer. Good? Okay. All right. And then you can also, and this is really, really common, not just with neurotransmitters, also with hormones and other chemical messengers. The other way you can manipulate the synapse is interfere with binding to the receptor, right? So just think about back to literature, and I'm going to tell you that there are 2 ways that you can interfere with binding to the receptor. You can have an antagonist, and an antagonist would sit in the receptor, and do you predict that it will cause a response or not? No. So it is basically a blocker, right? So if you want to block the effect on the postsynaptic cell, for instance, if you had excessive amounts of something and you couldn't get rid of the something, you can block the effect of the something by putting in an antagonist. Here's an example. Estrogen antagonists. So estrogen antagonists or estrogen blockers are used in a bunch of different situations. One of them is there are breast cancers that are susceptible to high levels of estrogen, and if you want to reduce growth in those breast cancers, you can give an estrogen blocker, right? Or what if I were a male to female transgender person, right? I would probably want to take an estrogen blocker. Does that make sense? So there are also testosterone blockers, and there are blockers for neurotransmitters. There are all different kinds. You've probably heard of beta blockers before. Those are all antagonists. So you can use an antagonist, or you can use an agonist. And an agonist sits in the receptor, and it's a mimic. So if the antagonist is a blocker, the agonist is a mimic. It sits in the receptor and causes a response that is similar to the chemical messenger. And you might use an agonist when you didn't have enough of something, right? Okay, so let's look at some examples of agonists and antagonists. Agonist drugs or chemicals produce a response that is similar to the normal activation. For instance, why do people like nicotine? It's always good to know why people like drugs before you start telling them that they shouldn't do them, because you really need to empathize before you know. So my children know entirely too much. I'm like, this is why people want to do this, and this is what happens. So why do people like nicotine? Part of the reason that people like nicotine is that nicotine is an acetylcholine agonist. Acetylcholine in the central nervous system increases short-term attention memory formation. So why do you like nicotine? especially the first few times people smoke. It's because it makes you feel really bright, right? You're like super with it for a second, okay? And people like that pleasant feeling. It's nice. Eventually, you also downregulate in response to it, and now you just have the chemical addiction without the mental acuity. But that's initially part of the reason that people like it. So nicotine is an acetylcholine agonist. It pretends like it's acetylcholine in the brain. And then, so antagonist drugs are chemicals. There's tons of them. Let's talk about a couple of them. Caffeine is your favorite. Well, not your favorite. But even in Utah, I know you guys drink caffeine. You might not drink it warm, but don't think I'm not noticing that you are. Okay, so caffeine is an interesting one. It's actually an antagonist, but get this. Caffeine is an antagonist to an inhibitory neurotransmitter. So can you see how it causes excitation? It's an antagonist to an inhibitory neurotransmitter. So it sits and blocks the activity of an inhibitory neurotransmitter, and therefore it's excitatory in your central nervous system. That's how it works. That's why it makes you feel alert. And of course, you downregulate with caffeine use as well. Not surprisingly, eventually, if you're using it regularly, it's not quite as effective as it was the first few times. Like, have you ever seen like the first time your kids try caffeine? like, oh my God, I wish I could get that again. It's just barely like making me conscious. That's all it's doing anymore. So that's how caffeine works. And then, does it surprise you at all that quite often poisons and toxins are antagonists? Yeah, especially if they're an antagonist with a greater affinity for the receptor than the actual ligand, then they can kill you dead, right? So let's talk about a couple of them. One of them is, fugu. I don't know if you guys have heard this, but fugu is a delicacy made from the pufferfish, okay? So the pufferfish are adorable little things that they will definitely tell you in just a second. Don't even try to eat me because you will regret it. But humans, we're really persistent, and we take it as a challenge. We're like, oh, really? Yeah? And so what fugu is they have a toxin that they produce, and this toxin is a defense mechanism. And when people eat fugu, it's generally a sushi, a delicacy, and you can't get it here. You can get it in California, and you can get it in Japan. I haven't tried it yet, but I'm going to. So even though when I tell you it's going to make me sound stupid, but at least I'm not bored. Like, I'm totally not bored. So what happens with fugu is it's got this bladder of toxin, and you pay like crap tons of money to get it prepared so that the fish won't kill you. But the toxin, you take out the bladder, and then a little bit of it has seeped into the flesh of the fish, right? It's supposed to be super delicious, but in addition to being super delicious, it also gives you a little tingle. That means you almost died. You weren't bored. So fugu is a neurotoxin, and what it does is it sits in your receptors for one of your neurotransmitters, and it has a greater affinity for the receptor than the actual neurotransmitter. I think it's acetylcholine, I can't remember. And it causes respiratory arrest while you're conscious. So yeah, that's what it does. But you weren't bored. Like half of adulthood is just figuring out what the hell to do with the next 40 years, honestly. What am I going to do? Is this all this thing does? So that's fugu. Has anybody ever tried it? I haven't met anybody who's tried it yet. Mostly it's going to be like super rich people, right? Okay. Next time I go overseas, I'm going to try it. Okay, my husband disagrees with that, but I'm just going to save up and then he can just witness it. Okay, so tetrodotoxin, and then saxitoxin. Have you guys heard of red tide? So red tide is this like protozoan bloom that happens in certain environmental conditions in the ocean. Yeah, in certain months of the year, in certain areas. And what happens is the protozoan produces a neurotoxin, and it gets into all of the fish and all of the seafood, and you can't go anywhere near anything. It like completely destroys the fishing economy while it's going on in those areas. It will kill you. Honeybee venom does the same thing. It's an antagonist, but it's generally just local, so it's not going to kill you. The same thing with sea anemone. Those things hurt. Have you ever stepped on one of those? They're a beast. They hurt really bad. And then black mamba venom, that's going to be your worst day ever because that's a neurotoxin and it generally does go systemic. So those are all antagonists. So you're going to end up with these kinds of situations. Black widow spider-venom does something similar. Go ahead.
Why is it that some of them are local and some aren't? Is it just are they broken down before they get in the bloodstream?
How much they're, usually it's how much they're released and whether you actually take them. If you just inject like honeybee venom just locally in small quantities, it doesn't get picked up enough to go systemic. But if you take something in large quantities, right, or if it gets injected into your bloodstream, right? Yeah, and also if it's lipid soluble, it's more likely to have brain impacts much more quickly. So it's got to do with quantity and chemistry. That's what it has to do.
You got to spend fewer times, but like maybe closer to your brain, like on your neck. That's just, you know, a few hours.
I doubt the honeybee venoms could do anything because of the venom. If you had an allergic response to it, it would be your allergies that killed you. I mean, theoretically, I suppose if you could just get a whole bunch of it in a syringe and then shoot somebody up, you might be able to kill them. Probably easier ways, but it's a good story. You're going to write that story. Go ahead. Yes, exactly. If it goes basically into your just connective tissues and relatively, just below the skin, it's unlikely to get picked up in large quantities. But I don't know. Probably somebody has died from honeybee venom. I don't know. Like Agatha Christie probably did it in one of her novels. I read all those as I was a kid. I know so many ways to kill people. I also took a forensics class when I was in college, and it was awesome. They called it Uncle Gil's story time because it was just like example after example of how to kill people, and like he taught us how to calculate how deep to bury the bodies based on temperature calculations and like how deep to sink them in the water. And he's like, the only reason people get caught is because criminals are lazy. And I was like, okay, so if you're really going to kill somebody, you just have to need to put some work into it. That's all. Okay, so clinical connections. I'm really not a sociopath. Okay, so clinical connections. Diseases can also do some of the same things. So let's talk about tetanus and botulism. Okay, so what does tetanus and botulism have to do with this story? Both of those are primarily in neuromuscular junctions, but they do sort of reinforce this story. So tetanus toxin is from the bacterium Clostridium tetani, which is a really common soil bacterium. So why doesn't everybody get tetanus? Well, first of all, vaccines are awesome. And then second, what happens is in order for Clostridium tetani to start producing the toxins that cause problems at your synapses, they need to get into anaerobic conditions. So how did your mom tell you get tetanus? Yeah, step on a rusty nail. And as usual, mom was at least part right? It doesn't have to be rusty, it doesn't have to be a nail, but it usually has to be a puncture wound that closes on itself. And when the puncture wound closes on itself, then you start to produce the toxin. And then even if you got antibiotics, it doesn't make any difference because it's the toxin, not the actual living organism that causes problems. So what it does is stops acetylcholine exocytosis at synapses for skeletal muscles. And so you end up getting skeletal muscles that cannot actually act properly. And if it happens enough, you can actually get paralysis of the diaphragm and it can kill you. So that's tetanus. And close to this one, is botulinum toxin. So this is from the bacterium, same genus, different species, Clostridium botulinum. Again, relatively common bacterium, only produces the toxin in anaerobic conditions. How'd your mom tell you you get botulism? Denicans, always part right. The problem is really that if you have something that you're canning, like let's say you're home canning food, what you need to do is to make sure that there are no living organisms in that before you create an anaerobic condition. So what are you supposed to do to the jar? Boil the hell out of it, boil the hell out of the lids, and boil the hell out of whatever you're going to can, and then you create the vacuum. So what's the deal with the dented cans? If there's organisms still living in there, then they'll generally produce carbon dioxide as a byproduct of their metabolism, which can make the can get distorted. So that little thing on the top, right? That little dimple on the top, that thing should be down, right? It's not a good indication if you open something and it hisses at you for like 20 seconds. You're like, oh, that is definitely going down the sink. So with Clostridium botulinum, this inhibits neurotransmitter release at skeletal muscles and it causes respiratory failure. And infants are incredibly sensitive to botulinum toxin. I don't eat home canned food unless I was involved in the canning. Pretty much don't. So you can give it to me at Christmas and I will write you a thank you note and then I'm going to pour it down the sink. Because I don't know. And so infants are especially susceptible to Clostridium botulinum. So that's botulinum toxin. And then black widow spider venom works pretty much the same as those two, but it usually stays local and doesn't go systemic. Okay, guys, questions? No? Everybody good? Okay, so I feel like I'm forgetting to tell you something. Okay, so I'm going to briefly start on neurotransmitters. You guys are a couple minutes ahead of the other class, so I'll probably let you go in a minute. But so what we're going to do with neurotransmitters to give you sort of a preview of how to organize, because you're going to feel overwhelmed at first. This is the first time that you're going to really have a dump of really unfamiliar material that requires both understanding and memorization. So what you're going to do with the neurotransmitters is we are going to organize them first by their chemistry because that's how they're categorized. Okay, most of them are based on amino acids. They are either a single amino acid. The biggest neurotransmitter is about 40 amino acids, but most of them are about the size of one. So they get categorized by their chemistry, like what amino acid are they based on, or are they big ones? And then after that, you are going to organize them based on sort of where they act. Are they a CNS neurotransmitter? You guys with me? Or a PNS neurotransmitter? Or both? Some of them work a couple of different places. So chemistry and then CNS or PNS. And then eventually you will start figuring out what their receptors are like. Okay, so what are the receptors called? Now, why on earth do you have to do this? Because what happens here is this is the point at which physiology and clinical applications start to have a dovetail. Have you ever looked at a PDR or a nurse's desk reference? And it says, for instance, this drug is a GABAergic antagonist, right? And works in the central nervous system. So what I'm going to try to get you guys comfortable with is what this sort of academic story has to do with sort of the clinical applications of the story. So you learn the chemistry of the neurotransmitter, you learn the basic functionality of the neurotransmitter, and then you learn the receptors for the neurotransmitter. And then whenever I think it's relevant or reinforces the story, I will tell you clinical connections with the neurotransmitter, which is usually where the drugs get involved because they're really interesting. The SSRIs, we'll talk about hallucinogenic drugs, we'll talk about methamphetamine, we'll talk about all that kind of stuff. And let me go ahead and show you something so that you don't feel overwhelmed. This table that's at the end of this set of notes right here is a table that, so I am obsessed with neurotransmitters. Like one of my hobbies, I know that you guys have cooler ones than I do, is that I have an entire spreadsheet that's like this big that is constantly developing about neurotransmitters and drug actions. So people ask me about this stuff, so I just made a subset for you guys so that you can, so when people want to know, what does heroin do? What does salvia do? What about THC? This is here for you because people ask me all these questions. I don't ask about any of this on the test unless it's also in the body of your notes. That's just a reference for you. And I have way more than this, but just people just really like that. So anyway, that is not something you need to worry about unless it's in the body of your notes, but it is a reference for you. So when you start wondering how ADHD drugs work and that kind of stuff. And then I also have a phenomenal book that I will bring for you guys next time and show you what it looks like, which is my Bible on neurophysiology. And half the material came from your textbook, but the other half came from that book. Okay, so we will stop there for today.